we are. Hello.
Hello, everybody.
Hello, everybody. Pleased to.
Hello.
Hello. Pleased to see you all here again this afternoon. As you know, this international seminar series on liver toxicity and steatotic liver disease try to encompass basic science with translational science. This afternoon, I have the great pleasure to present to you a leading expert in the field of drug-induced liver injury, who is Professor Gerd Kullak-Ublick. He is rather exceptional in the way that combines expertise in clinical pharmacology and also in pharmacovigilance. He is full professor of clinical pharmacology and toxicology at the University of Hospital Zurich, University of Zurich in Switzerland, but at the same time is a global head of mechanistic safety and safety signal management at Novartis in Basel, Switzerland. But also he combines all this knowledge with a background as clinical hepatologist. So really he's someone quite exceptional. And he will talk us about a very interesting project, the IMI TransBioline project. It's a consortium to develop liver safety biomarkers. He will, and he colleagues the DILI work package together with Professor Guru Eiffel. So thank you, Gerd, for accepting so nicely our invitation. You have the word.
Thank you very much, Maribel. So I will now, for the kind words, I will now try to share my screen. Okay. Please let me know if you can see the slide.
Yes.
Okay, good. Excellent. So thank you very much. Today's topic is about, as Maribel indicated, about liver safety biomarkers and the ongoing activities in the field of qualifying these new safety biomarkers. So I'll just go to the first slide. And many of you will be familiar with the topic of drug-induced liver injury, DILI, but just as a quick reminder, this is a leading cause of drug attrition in drug development and pharmaceutical industry, and the second most common reason for post-marketing withdrawals following cardiovascular causes. One of the key biomarkers, which many of you will be familiar with, is the so-called Hy's law, which is a combination of ALT, alanine aminotransferase, together with bilirubin. And Hy's law was originally coined by the FDA as a predictive marker indicating a risk for progression of DILI to severe liver injury, in other words, liver failure, liver transplantation, or death. So a patient who has a true Hy's law has a 10 to 15% risk of progressing to liver failure. And this law has been very reliable across numerous real-world databases, clinical trials databases, post-marketing pharmacovigilance databases. However, the problem is that sometimes it's really difficult to identify a true Hy's laws case. The lab values ALT and bilirubin can be confounded. For example, bilirubin can be elevated due to inhibition of a bilirubin transporter in hepatocytes, such as MRP2, which is the efflux pump for bilirubin into bile. If MRP2 is inhibited, you can find elevated bilirubin levels in blood. Or there may be other sources of hyperbilirubinemia, which are not due to hepatocyte injury, such as hemolysis, for example. And the other aspect is that the frequency of Hy's laws cases is low. This is true idiosyncratic liver injury is a rare event, 1 in 10,000 or less. And sometimes these cases are not seen during clinical development, phase 3 trials, phase 2 and phase 3 trials, and they only become apparent after marketing the drug, which then leads to sudden post-marketing withdrawals because of the high usage of the drug and the sudden onset of several cases of liver injury, which were not seen during development. So what we need is new biomarkers, which are more diagnostic, possibly more prognostic, which help us identify which patients really are at risk of progressing once DILI has been diagnosed. And these markers should be sensitive, specific, accurate, and precise, obviously, to allow early detection and also to really help decide whether a drug should be discontinued or not. So there is a clear definition of what a biomarker is in FDA terms. FDA uses the so-called BEST category or BEST definition, which is a resource. The abbreviation stands for biomarkers, endpoints, and other tools. And this BEST definition essentially defines different categories of biomarkers. I just want to pick out 3 of these maybe in the context of liver safety. One is the category diagnostic biomarker. The second is prognostic biomarker, and the third is safety biomarker, because these are the 3 categories where we could categorize new liver safety biomarkers. So let's say the lowest level of evidence would be required for a simple safety biomarker, which essentially just tells you whether you have a safety issue with a given drug as characterized by a specific lab value or a new protein in blood, which is elevated. A diagnostic biomarker requires a little bit more evidence. It basically confirms the presence or absence of disease. So this would be more of a disease-specific biomarker. And finally, a prognostic biomarker is a biomarker that really helps you identify the subsequent clinical course of the patient, whether the patient is likely to progress to more severe liver injury or not. And obviously for that, you need quite a high level of evidence to really coin a biomarker as being a prognostic biomarker. So just moving on. So one of the aims of finding new biomarkers or to be able to use a new biomarker on a routine basis, biomarkers need to be qualified with a regulatory authority such as FDA. And a qualification is a structured process within regulatory agencies such as FDA or EMA, the European Medicines Agency. And one of the key things that you need is a so-called context of use for the biomarker. In other words, an exact description of under which circumstances the biomarker can be used. When is it really useful? So the context of use is very important. And so the kinds of contexts of use that you could envisage are situations that help you to define inclusion or exclusion criteria, to define into which treatment arm to allocate a patient. These would be typical contexts of use for use of biomarkers, whether or not to stop treatment during a clinical trial, support dosage selection, etc., etc. So, and even treatment response. So these are examples of contexts of use. So it's very critical in the whole qualification process to define your intended context of use at the start of your clinical study. So this is not a fishing exercise. You do not treat several hundred DILI patients and then see what do you have. What could the biomarker be useful for? You really have to think at the beginning, which is the context of use that is most that is needed most, into which I could fit my new biomarker. So these are the just 2 processes, EMA and FDA. Now, without going into too much detail here, you can see these are complex processes that extend over several months or even years, depending on the the amount of data that needs to be provided. The FDA process is slightly simpler. You start off with a so-called letter of intent. This is something which you submit at the beginning, at the outset, which describes which biomarkers you plan to investigate, what the context of use is within which you want to use these biomarkers, and based on outcome, this would then allow you to qualify a biomarker for that context of use. So we submitted, I will go into this in just a moment, in the consortium, which we are currently working in, the Transbioline Consortium. We submitted a letter of intent to FDA. This was reviewed by FDA, and FDA then issues what is called a determination letter, which either supports or refutes further investigation according to the contents of your letter of intent, and they provide recommendations of what else to take into consideration, possible changes of the context of use, etc. And then the next step is to submit the full qualification plan, the BQP, the biomarker qualification plan. This is already at a fairly advanced stage of the of biomarker assessment, when you have quite a concrete idea of what the biomarkers can do, how they can perform, in which context of use. You can still modify the context of use between the letter of intent and the qualification plan, but what you submit with the qualification plan is binding. That is basically the final protocol, if you like, which will then also be used as a basis for FDA to decide whether the level of evidence that you plan to provide will be sufficient, whether the context of use will be of interest and can be supported by your experimental plan that you describe in detail in the qualification plan. And then following generation of all the required clinical data, you submit the final package that contains all the data, and this is the so-called full qualification package, and FDA then reviews this, and if all is well at the end of that, the biomarker can be qualified for the said context of use. So the framework within which we are conducting these studies is a public-private partnership funded by the Innovative Medicines Initiative, which is actually the biggest public-private partnership in the health sector, and IMI, which has now been succeeded by the IHI, the Innovative Health Initiative. This is the IMI2 program, which funded the Transbioline Consortium, which is now in its 4th year. Transbioline started off in February 2019, so it's actually in its 5th year, and consists of a number of academic groups, as shown at the top here, the so-called applicant consortium, consisting of academic groups as well as small and medium enterprises. These groups are recipients of funding from the European Union, and then we have FPA partners shown here, which contribute funding in the form of in-kind contributions, as well as scientific and academic experts. This can include regulators as well as advisors. So this is the constitution of the consortium. The academic lead of the whole consortium is with the University of Zurich, and the industry lead is with Pfizer. But within the DILI work package, we have 5 different organ work packages, as shown on the left here: renal, liver, pancreatic, vascular, and neural injury. The liver injury work package is headed by Professor Guru Aithal from Nottingham University and myself. So where do we stand at this point in time? We have completed the so-called learning phase, which involves recruiting 100 DILI patients. And we are currently now in the confirmatory phase, which involves recruitment of another 200 DILI patients. And you can see the academic centers that are recruiting in this table here: universities of Nottingham, Zurich, Munich, Malaga, Reykjavik, Gothenburg, and Bern. and the number of DILI cases that have been recruited by each center, as well as essentially as controls, cases of acute liver injury not caused by drugs, and we call these non-DILI acute liver injury cases. So, for example, hypoxic liver damage would be an example for that. And the way this works is once a DILI patient has been recruited by an academic center, the case will typically be, first of all, recruited into the database of the consortium. Blood is taken for measurement of the biomarkers shown on this slide, and then the case is subsequently presented to an expert panel of adjudicators, and then the case is adjudicated as being either DILI or non-DILI. So this is essentially expert opinion-based case adjudication to see if the case is true DILI or if it is a case of non-DILI acute liver injury. And the biomarkers we are studying are shown on this slide here. So we have basically, this is just an overview here. In this cartoon, you can see some standard liver labs, ALT, AST, INR, and total bilirubin. These are the traditional biomarkers. And new biomarkers we're looking at are markers of cell injury, cell necrosis, such as HMGB1, which is high mobility group box 1, keratin 18, which is a cytoskeletal protein in hepatocytes, but in many other cell types as well, a caspase cleaved form of keratin 18 called CCK18, caspase cleavage being a marker of apoptosis, which is generally considered to be a prognostically favorable situation in the setting of acute liver injury, as opposed to pure necrosis. MicroRNA 122, which is a hepatocyte-specific microRNA. More than 50% of the hepatocytes microRNA is microRNA 122. And finally, GLDH, which is glutamate dehydrogenase, a marker of mitochondrial injury. We have some immune markers, which we also look at. One is osteopontin, which is a cytokine, typically elevated in cases of severe liver injury, as well as a macrophage marker, macrophage colony stimulating factor receptor 1. This is expressed on Kupffer cells and macrophages and is shed and measurable in blood in cases of immune activation, such as in idiosyncratic DILI. So these are the markers that are being studied. We're also assessing the MELT score, which additionally also includes serum creatinine, as well as bilirubin and INR as a marker of liver function. So these are the basically 5 patient groups, or 5 which have been compared with each other. On the far right, you can see healthy volunteers. On the far left, actual patients with drug-induced liver injury. Criteria here are lab values, as shown here, an ALT of 5-fold or more, the upper limit of normal, and or an ALP, an alkaline phosphatase, of more than 2-fold the upper limit of normal, or the so-called Hy's Law constellation, which is an ALT of 3-fold, combined with a total bilirubin of 2-fold the upper limit of normal. Other causes of liver injury should be excluded. This is the task of the expert adjudication panel to make sure no other causes of liver injury were present. And if the case is adjudicated as being DILI, the patient can be allocated into the DILI arm here. Then we have patients with acute liver injury of non-drug cause, similar inclusion criteria. However, as assessed by the adjudication committee, there are other confounding causes of liver injury which do not make the case suitable as a DILI case, and obviously chronic disease should also be excluded in these patients. Then we have patients with chronic liver disease. We have alcohol-related liver disease, as well as NAFLD as examples of chronic liver diseases, as well as patients receiving methotrexate as a hepatotoxic drug. These are patients with rheumatoid arthritis or psoriasis, which by definition would lead more to a chronic type of DILI. As part of the qualification process, the lab assays that are performed, these are performed by a biomarker company in Reutlingen, Germany, called Steatotic. The biomarker qualification requires very strict validation procedures, and these have to be adhered to by the lab measuring the biomarkers. We essentially have two platforms which we use. One is an immunoaffinity LC-MS platform. The other is sandwich immunoassays in a monoplex format. And without going into details here, things to consider are reference standards, obviously the matrix, which is EDTA plasma in all cases, easy to obtain. Quality control measures have to be performed. The accuracy and the precision of the assay has to be measured in multiple runs with acceptable accuracy at 80 to 120%. Stability evaluation, in other words, whether the sample can survive repeated freeze-thaw cycles. What is the temperature at which the sample has to be stored? Is it -20, -80? These are important quality parameters that have to be looked at. Selectivity, reproducibility, parallelism, and interference with, which goes into specificity. All of these things have to be provided in the so-called validation plan. There are three regulatory documents, one issued by the Critical Path Institute, one by FDA, and one ICH guideline, which essentially are used for putting together the qualification plan, the validation plan for the biomarkers. So this is very important. Okay, so then moving to the actual context of use, which is, as mentioned, a very important part of the whole qualification procedure. So the context of use which we have defined for DILI biomarkers is as follows. A single biomarker or a combination of biomarkers will aid in identifying patients with acute liver injury suspected to be caused by drugs without underlying liver disease, in whom the degree of liver injury suggests an increased risk for severe outcome, i.e. liver failure, transplant, or death, and dose reduction or dose interruption is warranted. Now, this is important because the FDA guidance for industry suggests interrupting treatment if ALT reaches the 8-fold threshold. So an 8-fold ALT leads to a recommendation to interrupt or even permanently discontinue drug treatment. Alternatively, an ALT of 5-fold for at least 2 weeks or more. And obviously, these are very non-specific thresholds and recommendations through lack of having better markers, and therefore understandable. But the aim here would be to complement ALT measurements with more specific biomarkers that actually provide more information into the risk for progression than ALT can provide. So in the so-called learning phase, as mentioned, we had 100 DILI patients in the learning phase, 40 patients with non-drug acute liver injury, 50 healthy volunteers, 53 patients with NAFLD or alcoholic liver disease, and 29 patients receiving methotrexate. And these are just the normal ranges for the biomarkers that are being studied, shown here. This is part of the biomarker validation. Just moving on to the next slide. This is the distribution of injury phenotypes between the DILI patient set and the non-DILI acute liver injury patient set. And you can see roughly similar distributions, 14% cholestatic injury in DILI, 15% in non-DILI, 65% hepatocellular injury, which is the most common form, and 21% mixed type injury. Severity at initial DILI diagnosis, 44% mild, 43% moderate, 13% severe. And outcomes, obviously these are outcomes based on these included patients here. Any form of progression was seen in 15 patients. Of these, 12 either had liver transplant or fatal, and 3 had an unknown follow-up. And these numbers are essentially similar in the non-DILI acute liver injury group. So just coming to the first set of results here for biomarkers, you will see box plots here for the different biomarkers. So GLDH, we have 2, one for GLDH, protein levels in blood, the other for GLDH activity. So GLDH, keratin 18, caspase cleaved keratin 18, HMGB1, osteopontin, MCSF1R, and ALT. And in each box plot, you have 5 different groups: DILI patients, acute liver injury non-DILI, NAFLD or alcoholic disease, methotrexate patients, and healthy volunteers. Note the logarithmic y-axis here. And so there's just the message from this slide is basically DILI and non-DILI acute liver injury had high levels, significantly high levels of each biomarker compared to the other groups. So that was the first finding, and just refining this a little bit, taking out the other patient groups. This is now DILI compared to acute liver injury of non-drug causes compared to healthy volunteers. And again, you can see, I mean, the first finding is that GLDH, both protein levels and enzyme activity, shows a similar pattern, which is good news. Again, these 2 patient groups were obviously higher than healthy volunteers. And just moving on to the comparison of DILI versus non-DILI. Here you can see, if you take CCK18, for example, there was a tendency towards slightly lower levels in DILI compared to non-drug acute liver injury. Same applies to keratin 18 as well as to MCSF1R here. Not so for HMGB1 and osteopontin. And these are ALT levels here, where actually the same pattern was seen, slightly lower levels in the DILI group compared to acute liver injury. So this table shows the ROC. So this is the diagnostic performance of each biomarker to separate DILI from non-DILI injury. So this is acute liver injury versus drug-induced liver injury. And you can see ALT was not useful for discriminating the 2 groups. Neither were any of the other biomarkers. So what these biomarkers cannot do is discriminate between drug-induced liver injury and other forms of acute liver injury, because an AOC of at least 0.75 would be required to make the finding significant. That was not the case for any of these markers. However, now we look at patients who progressed to severe liver injury. 15 patients, if you remember, were so-called progressors, compared to patients who did not progress. So yes indicates progressor, no indicates non-progressor. And you can see, for example, HMGB1 down here, bottom left, the progressors had higher levels compared to non-progressors. This was at the initial time point of DILI, not at the time of progression, but before progression occurred. So at a point in time when you would not have been able to predict progression based on the standard liver lab values. HMGB1, the same was seen for keratin 18, higher levels in progressors, also for MCSF1R as well as for osteopontin. So 4 biomarkers tended to have higher levels in subsequent progressors compared to non-progressors. And just to show you which patients progressed, patients who were initially presented with mild liver injury, 44 patients. 5 of these progressed either to moderate or severe or transplantation. Of the 43 patients who presented with moderate injury, 4 patients progressed, and the severe patients, 13, had 6 of these had transplantation. Now, in the setting of clinical trials, the kind of patient you're going to see is the patient with mild DILI, not necessarily in clinical routine where you see all phenotypes, but this is your typical clinical trials patient, the kind of patient who nowadays would have treatment interruption if ALT reaches 8-fold threshold. These patients are what we see, and patients who have 8-fold ALTs all have interruption of drug treatment, and this is one of the big problems in drug development. Too many patients are being stopped because of an ALT of 8-fold in this mild DILI group. Just moving on here. So this is now the ROC or AUC value of progression versus no progression, and this is obviously of interest now. So an ROC or AUC of at least 0.75 would be a significant value. Of interest, the MELT score and albumin were among the best predictors of progression. MELT score, of course, also contains creatinine, bilirubin, and INR. Albumin was also a good predictor, as was total bilirubin, which is part of the Hy's law algorithm. Non-predictive was ALT. MCSF-R1 came close to the 0.75 threshold. 0.78 was seen for HNGB1, and osteopontin was also close to the threshold. So HNGB1, osteopontin, and MCSF-R1, at least in the current analysis, are the 3 most promising markers for prediction of progression. If we zoom in only on patients with mild injury, as mentioned previously, these are the ones we typically see in drug development in pharmaceutical industry. These are the relevant cases for clinical trials. Here we had a total of 5 progressors compared to 39 non-progressors. So we're comparing 5 patients with 39 patients here. And you can see there was marginally higher HMGB1, bottom left panel. There was a trend for higher results in progressors, also for K18, for CCK18, as well as MCSF1R. Of course, these are small sample sizes, and we would need bigger sample sizes to maybe see significance. But the trend was there. So again, these are the ROC or AUC values just for the patients with mild injury. And again, MELT and total bilirubin were the best scorers here. But HMGB1 was also good with 0.77, and MCSF1R came close to the threshold. This is the final slide here, final data slide. This is essentially a scatter plot, which shows it's a complicated slide, but if you just maybe focus on these curves shown in the central part of the plot, the blue curve, this is these are essentially the findings for patients who progress, so-called progressors. And behind it, the red curve, these are the non-progressors. And you can see there was a substantial overlap between progressors and non-progressors, but we do see some markers where the peaks do not overlap, and these are obviously the ones which could be most promising. Bilirubin is shown here, for example, and MCSF1R is here, and HNGB1, and these are obviously the biomarkers which show biggest promise for being able to differentiate progressors from non-progressors. So just in summary, Biomarkers are an essential tool for drug development, significantly contributing to drug safety and patient safety. An efficient pathway to facilitate development of biomarkers is regulatory qualification, which can improve drug safety, can save costs, and reduce development times. A major challenge with DILI currently is the lack of adequately sensitive and specific diagnostic, safety, and prognostic biomarkers. And for this reason, the IMI Transbioline Consortium set out to qualify new biomarkers, being a big public-private partnership between industry, biotech, academia, and regulators. We have encouraging results in terms of prognostic performance from the initial learning phase, and now aim to confirm this in the upcoming confirmatory phase, and hope at the end of the initiative to be able to qualify at least 1 or 2 new liver safety biomarkers as predictive markers of liver injury. And I would like to express my big thanks here to Michael Merz, who is running the regulatory qualification within the consortium, Oliver Pötz from the company Steatotic, developing biomarkers, and to the Pfizer colleagues who did all the statistical analysis, Dean Lee, and who provided all the box plots shown in today's presentation. Thank you very much.
Thank you very much, Gerd, for this excellent overview of where we are standing right now in terms of biomarkers development. It's a long effort that we have been accomplished through the years, and now we are, well, as you said, showing quite promising results in terms of prognostic biomarkers. I would like to open this section for discussion. Is there anyone who wants to make a Gerd Javier Cubero, who is leading this also these seminars, has to leave because he has another appointment.
Yes, Gerd, congratulations, fantastic update of the biomarker plan for validation and all the efforts made in this year. And my question is with regard to drug-induced liver injury expressed with a wide range of phenotypes. Sometimes it's difficult to accept that for one phenotype very different from another, the biomarker is going to perform similarly. What do you think about that?
Right, absolutely. That would be the next step, of course, if we can use these biomarkers to differentiate between different phenotypes of DILI. Put it this way: if we take classic idiosyncratic DILI, which is an immune-mediated process, as maybe a standalone category of DILI. For that, we have found that MCSF1R, for example, which is an immune marker, has highest levels compared to other biomarkers. It was originally actually identified by AstraZeneca when they were developing Simulagatran, a thrombin inhibitor, which was not marketed because of too many liver injury cases in the phase 3 trials. But it was a useful exercise because they performed metabolomics and found that MCSF1R was a biomarker with a high blood level in patients with Simulagatran liver injury. And we actually, in the previous IMI consortium, which had a similar mission to qualify new biomarkers before TransBioline. This was called SAFE-T. In the SAFE-T consortium, we had a number of patients who were taking a drug which was then withdrawn from the European market, EMA, and this was a central muscle relaxant. And there again, we had very high levels of MCSFR1 in these very idiosyncratic forms of liver injury. So at least we can say maybe immune markers are useful for idiosyncratic liver injury, but obviously there's going to be a lot more to look at. We have these mixed causes of liver injury, immune attack, plus some intrinsic mechanisms, reactive metabolite formation, inhibition of BSEP, the bile acid transporter, mitochondrial toxicity, whatever. All these things could also have biomarkers which indicate the underlying mechanism of injury. GLDH could be a marker of mitochondrial liver injury, but this will have to be looked at in subsequent analyses.
Thank you.
Thank you. Thank you, Raul. Yes, I think this is a work on progress to new data is evolving. Gerd is also his special interest in transporters. He has already make some comments regarding biliary transporters. So what do you think also about the possibility of bile acid salts being measured in drug-induced liver injury?
Yes, very good question. We do actually have bile acids in TransBioline as well as biomarkers, and you know, bile acids have been looked at over decades now for various kinds of liver disease, and the overall conclusion was they have not been particularly useful in diagnosing any specific kind of liver injury. I mean, they are markers of cholestatic liver injury in intrahepatic cholestasis of pregnancy. They're useful for assessing the severity of ICP. 10% of pregnant women have elevated serum bile acids in the last trimester of pregnancy due to hormonal effects on bile acid transport. So these are interesting leads, but they were never considered to be useful beyond intrahepatic cholestasis of pregnancy. Within TransBioline, we have been measuring biomarkers. We have seen a few biomarker species which definitely are strongly elevated, even in pure hepatocellular idiosyncratic liver injury. So these are not just markers of cholestasis. What exactly that means will have to be looked at, but maybe I can share this much with you. We recently had to interrupt a clinical trial at Novartis because of liver injury, and we found a particular bile acid species which was elevated in these patients at baseline. So this was a bile acid which was elevated at baseline and predicted the subsequent onset of liver injury in these patients, obviously just with this one drug.
Yeah.
One would have to see whether this is reproducible in other forms of liver injury, but this, you know, there's probably more in the whole bile acid field than we think. Bile acid metabolism is highly complex with numerous involved enzymes, and there will not be an easy answer to this, but I think it's worth looking into this in more detail to really see for what could bile acids be useful.
Yes, for sure. Yes, that's very interesting. Any other question from the audience? Yes, Anna. Anna Licata.
Ciao,
from
ciao.
Italy.
Hi.
Wow, she's leaving. I thought she was going to make a question.
Is mute.
Oh,
Anna is mute.
I'm sorry.
Okay.
Yes, you can hear me? Yes?
Yes,
Okay.
perfect.
I saw a little difference between progressors and non-progressors around markers. And how do you measure progressors? By fibroscan, by liver biopsy, clinically?
Yeah, good question. So it's basically a patient who comes with mild liver injury, which would be defined as an ALT of more than 5-fold. If that patient goes on to develop a bilirubin of more than 2-fold, that would be considered progression. So if he progresses to a Hy's law situation from mild to Hy's law, that would be considered progression. A patient already has Hy's law and goes on to develop liver failure or transplant or death, that would also be progression. So it's a combination of lab parameters and clinical outcome, no imaging.
So progression doesn't mean chronicization. It's okay?
No, that's correct.
It's progression in liver injury.
Exactly, yes.
Okay.
In the
Okay.
acute episode, that's right.
In the
Yeah.
acute
Okay,
episode.
okay.
Yes. Hello,
Thank you.
Bernard. Bernard from ET.
Yes,
Yes, you got-
very interesting presentation. However, for the mitochondrial marker, the GLDH, it is somehow disappointing, and it tallies with former studies about, related to this marker. So I wonder whether in the future your consortium will consider other markers of mitochondrial dysfunction. And I am thinking about GDF15 and FGF21. These markers are elevated in some diseases, in particular hepatic disease, where there are some mitochondrial dysfunctions. So I don't know whether it is it will be interesting to study such markers. And do you have any data on this issue or not? Thank you for the
Actually,
answer.
not. Thank you. Yeah, not on those specific markers that you mentioned, but these are absolutely, and this goes back to Raul's question as well. This will be the next step to have biomarkers that really indicate mechanism of injury, especially these intrinsic mechanisms like mitochondrial injury. So that's definitely something we would probably have to include in a next initiative, trying to get these mechanism-specific biomarkers included as well.
Thank you.
Thank you. Gerd, in the study that we did with comparing biomarkers in drug-induced liver injury patients, whole cohort, and non-acute drug-induced liver patients, mainly viral hepatitis, we found osteopontin as a good marker of those patients that went on to a worst outcome. It was it has very distinctive rock area under the curve, similar to the MELT or even INR or albumin. So I wonder if the not so impressive results with osteopontin might be due to the type of procedure that you follow for the identification of osteopontin, of the variability that you may have with the technique. Could you tell us a little bit on that?
Yeah, it's a good question, and that was actually a bit surprising because,
yes.
as you say, the studies you mentioned showed a good performance, and actually preclinical studies have also been useful here in rodents. Osteopontin is strongly elevated in models of severe liver injury, and it's a cytokine. I think possibly if we had bigger groups of patients, we might see more significance. This may be related to the way this is now powered statistically, that the small patient groups. I think it's probably among the 3 or 4 better biomarkers for determining prognosis, but yes, but performance wasn't quite as good as expected. Yeah, good question. I'm not.
Yeah.
I think we might just need more patients.
Might. Are you considering the possibility also of seeing those progressors in non-acute, non-DILI acute liver injury patients to see if you find the same results that for DILI?
Finding, comparing.
Comparing non-DILI acute liver injury, those progress.
Oh, I see. Yeah, okay, yeah. Yes, good
Maybe
question.
You do not.
Yeah, I don't think that analysis has been done yet,
no.
but that's a good question. Yeah.
Maybe.
So progression in non-drug acute liver injury. Yeah. Yeah.
Yes. Well, there are many questions on this. Bernard, do you want to talk again?
Yes,
perfect.
I'm again. Yeah.
Yes,
I may have missed something, but I don't remember whether you mentioned whether the patient had a liver biopsy for histology.
Not as part of the transbioline. It is possible that a few patients had biopsy just as part of the clinical management, but this was not a protocol requirement.
A protocol,
yeah.
Okay. Okay.
So yeah, but unfortunately,
no. It's. I mean, you did a great job. I mean, it would have been much harder.
Yeah. Right.
Thank you.
Well, if we are about to perfectly in time, if there is no more questions, we could go on talking with you, Gerd. But I think you have illustrated us very well where we stand now on biomarkers development and qualification. And we hope to see the results of this confirmatory phase and to see what we learned from the results of this very large population of DILI patients, indeed, because this has been a very important achievement of this consortium. The great number of samples that have been collected in relatively rare disorders as DILI. So this has been and the qualification of all of the adjudication of all these samples. Thank you very much, Gerd, for being with us this afternoon. We hope to see you soon.
Thank you very much for the attention.
Thank you.
Thank you for your talk.
All. We will next month we will have a new telecom. The last conference in this first 6 months. So we expect your participation in this new conference at the International Seminar Series. All the best to all of you. Thank you for attending this meeting. Bye.
Thank you.
Bye.
Thank
Bye.
you, Isabel.
See you in Milan.
Yes.
See you.
Bye.
See you. Bye.